Welcome to Bio Bits and Bytes. Today, we're diving deep into something really fundamental. How a cell decides what it wants to be when it grows up, so to speak. Our focus is on stem cell development and a particularly fascinating molecule called alpha-ketoglutarate, or AKG. You might have heard some buzz around it, sometimes even called a bit of a Marvel metabolite.
It really is. It's a small molecule, chemically speaking, but its impact, particularly in those very first stages of development, is quite significant.
Exactly. And to really help us and you get our heads around this, we've gathered some fantastic resources today. We'll be looking closely at a research article from MB Press. That's the one that first really shone a light on AKG's potential magic.
That titular paper really kicked things off in some ways.
Right. We also have a great overview from Wikipedia on epigenetics in stem cell differentiation. Crucial background. Then we'll explore a piece from PMC giving us a broader view on metabolic changes in stem cells. And finally, we'll unpack a very recent, really exciting study from Nature Cell Biology that looks specifically at what AKG does in human embryonic stem cells. So quite the journey ahead.
It is, and each source gives us a different angle, doesn't it? The MBO paper sparked the interest. Wikipedia provides that epigenetic framework.
The PMC article zooms out to the bigger metabolic picture.
And Nature Cell Biology brings in the latest crucial human data. It pieces the puzzle together.
So our mission today is to understand how this seemingly simple molecule, AKG, plays such a pivotal role, how it influences the destiny of cells right at the start of life. We've got our expert here to guide us through the science. And don't worry, we'll be asking for clear explanations of the technical terms, breaking things down so everyone can follow this microscopic journey. Okay, let's dive right in then. First things first. What exactly is alpha-ketoglutarate? And why the magic label, especially regarding early germ cell development?
Okay, so alpha-ketoglutarate, AKG, is fundamentally a really central molecule in how our cells generate energy. It's a key intermediate in what's called the Krebs cycle or the citric acid cycle. You probably encountered that in biology class.
Well, the cell's engine room.
Exactly. But what makes it so interesting for stem cells isn't just its role in energy production, it's also a crucial cofactor.
Cofactor. Oh, we can break that down for us.
Think of it like a helper molecule. Certain enzymes, which are proteins that carry out chemical reactions, need a cofactor to function properly. And AKG is essential for specific enzymes that modify the epigenome, they influence how genes are read, and ultimately control the cell's identity.
Ah, okay. So it enables these other important molecular machines.
Precisely. The 2019 study by Tischler and colleagues really brought this to light in mouse embryonic stem cells, or MESCs. They found AKG helps maintain pluripotency.
Pluripotency, that's the ability of these early cells to become basically any cell type in the body, right?
That's exactly right, the amazing potential stem cells. But what was also striking was that AKG enhanced the differentiation of these MESCs into primordial germ cell-like cells, PGCLCs. These are the precursors to sperm and eggs.
So it helps them stay flexible and helps them become germ cells. That sounds a bit contradictory.
Well, it points to its nuanced role. It's not just fuel. It seems to be actively involved in guiding these crucial cell fate decisions. That's where the magic idea comes from, its ability to influence these fundamental processes.
Okay, that makes sense. It's more than just energy. Now, the MBO Press article also mentioned some interesting metabolic shifts during these early stages. Can you tell us more about that? What's happening with the cell's energy usage?
Certainly. This refers back to a 2017 study by Hayashi and colleagues. They observed that as mouse embryonic stem cells transition towards a slightly more developed state called epiblast-like cells, or epiLCs, their main mode of energy production changes.
Changes how?
They shift away from using oxidative phosphorylation, or OXPHOS, which is the really efficient oxygen-dependent way to make energy in mitochondria, and they start relying more heavily on glycolysis. Glycolysis is faster, but much less efficient, and it doesn't require oxygen.
Okay, so they switch energy strategies. Why?
That's the big question. But then, interestingly, as these epiLCs develop into those primordial germ cell-like cells, the PGCLCs, the pattern reverses again. They shift back towards using OXPHOS and become less reliant on glycolysis.
Wow, so it's like they're strategically flipping metabolic switches as they progress.
It certainly seems that way. The cell is actively changing its power source. And as we'll see, this metabolic state is really tightly linked to the cell's identity and fate.
Fascinating. And this is where AKG comes back in. What happened when Tischler's team actually added extra AKG to these mouse cells?
The results were pretty dramatic. Adding AKG seemed to favor the self-renewal of the MESCs. It helped them stay in that pluripotent state delaying differentiation.
Okay, so it supported the stay flexible option.
Yes. But even more striking was the effect on PGCLC production. Just by adding AKG, they saw roughly a 50% increase in the number of PGCLCs formed.
A 50% jump, just from adding this one molecule.
It was a significant boost, and the study even suggested that AKG might potentially be able to replace some of the complex, expensive cocktails of growth factors usually needed in the lab, things like the 2-lift medium for pluripotency or BMP signaling molecules for PGCLC induction.
Replacing entire mixes with one metabolite. Okay, now I'm really seeing the magic angle for lab work at least. Now, you mentioned a Kg is a cofactor for enzymes modifying the epigenome. The research specifically mentions histone and DNA demethylases. That sounds complicated. Can you unpack that for us? Why is it so important?
Absolutely. This gets us into epigenetics, which that Wikipedia overview covers well. Simply put, epigenetics involves changes to gene activity that don't change the underlying DNA sequence. Think of it like bookmarks or sticky notes on a book's pages.
Okay, annotations that change how you read the book.
Exactly. Histone and DNA methylation are key types of these epigenetic marks. Methylation involves adding a small chemical tag, a methyl group, to DNA, or to the histone proteins that package DNA. Generally, methylation acts like a silencer or a dimmer switch for genes. It tends to turn gene activity down.
So it quiets things down. And demethylases.
Demethylases do the opposite. They remove those methyl tags. And AKG is absolutely essential as a cofactor, a helper for two major families of these enzymes. The JMJC domain containing histone demethylases, or GHDMs, and the TIT DNA demethylases.
So AKG enables the eraser enzymes that remove the silencing marks.
Precisely. By enabling these demethylases, AKG directly influences which genes are active or inactive. It shapes the entire epigenetic landscape of the cell. And as the Wikipedia piece points out, this epigenetic landscape is fundamental in determining what a stem cell becomes. So AKG isn't flipping the switches itself, but it's providing the power for the enzymes that do.
That clarifies it beautifully. It's a crucial enabler for reading the genetic blueprint correctly for a specific cell type. Now, the MBO article also threw a curveball. It mentioned that a modified form, dimethylation or DMETG, which gets into cells easily, might have other effects non-epigenome activities. What are those and how do they, well, complicate things?
Yes, DAMAG. It's a useful tool in research because it crosses cell membranes better than regular AKG, but it definitely adds layers of complexity. Biology is rarely straightforward.
Tell me about it.
Well, for example, a 2014 study by Howe and colleagues found DMEK can stabilize a protein called HIM-1A.
HF1 sounds familiar. Hypoxia, low oxygen.
Exactly. HI1 is the master regulator of the cell's response to low oxygen. By stabilizing it, DMAKG can basically trick the cell into thinking it's hypoxic, even if oxygen levels are normal. This triggers a whole set of gene expression changes related to hypoxia.
So it's sending a false signal about oxygen levels, unrelated to the demethylase activity.
It seems so. And there's more. Some studies, like one by Chin et al. In 2014, suggested AKG might inhibit ATP synthase, part of the core energy production machinery.
Wait, inhibit energy production? That seems counterintuitive.
It does. And that study also suggested it might inhibit mTOR, a key sensor of nutrients and regulator of cell growth. However, as the EMEO article notes, other research, like a 2016 study by Tesla et al., found conflicting evidence they didn't see that effect on ATP synthase with added AKG. So the jury is still out on some of these effects.
Okay, so conflicting data there. Anything else?
Yes, one more piece from a 2015 study by Carey et al. They observed that treating naive mouse ESCs with DMAKG led to changes in specific histone methylation marks, H3K9Me3 and H3K27.3, which are generally repressive marks. These changes were linked to maintaining pluripotency. So it points back to epigenetics, but maybe through different pathways too.
So using DMAG means we have to be really careful interpreting results because it could be doing several different things at once. Stabilizing HIF, maybe affecting energy, and influencing methylation.
Precisely. It's a powerful tool, but not a perfectly clean one. It highlights the need for careful experimental design.
Right. Okay, let's zoom out a bit. The PMC article talks about metabolic reprogramming in stem cells more broadly. How does this specific story of AKG fit into that larger picture of stem cell energy and identity?
The PMC article gives great context. It emphasizes that different stem cell types often have distinct metabolic signatures. For instance, primed pluripotent stem cells, which are a bit further along developmentally than the naive ones we've mostly discussed, tend to rely heavily on glycolysis. even when there's plenty of oxygen.
Glycolysis, that's the less efficient pathway. That sounds like the Warburg effect seen in cancer cells, doesn't it?
Very much so. It's a strong parallel. Studies by Vorum, Zhu, and others really established this reliance on glycolysis in primed PSCs, the hallmark.
And what about other stem cells, like adult stem cells?
Adult stem cells which reside in our tissues are often quiescent, sort of dormant, waiting to be activated. Their metabolic state seems different again, often characterized by lower metabolic activity, perhaps relying more on pathways like fatty acid oxidation, though it's complex and still being actively researched.
So different states, different energy sources. How does EKG tie back in?
The crucial link, highlighted by both the PMC and MBO pieces, is the connection between these metabolic pathways and the activity of those epigenetic enzymes we discussed. The availability of key metabolites like AKG, but also others like acetyl-CoA, directly fuels or limits the enzymes that add or remove epigenetic marks.
So the cell's energy status directly impacts its ability to rewrite its own instructions.
Exactly. Metabolism isn't just housekeeping. It's an active layer of regulation that's deeply intertwined with cell fate decisions. What a cell eats, or how it makes energy, influences what it can become.
It's all connected: metabolism, epigenetics, cell identity.
Wow. Now, let's shift to that very recent Nature Cell Biology paper. This one focused specifically on human-naive embryonic stem cells, or NESCs, a different system. What did they find about AKG's role in making trophectoderm? That's the tissue that forms the placenta, right? Yes, the trophectoderm TE forms the embryonic part of the placenta, absolutely essential for pregnancy. And this study by Van Arum and colleagues found something really interesting and actually quite different from the mouse findings in some ways.
Different how?
They saw that when you induce human-naive ESCs to become trophectoderm, the levels of IKG inside the cells naturally increase. They accumulate it. And crucially, if they artificially increased IKG levels, it potentiated the ability of these human cells to commit to that trophectoderm lineage.
So in humans, higher IKG seems to push them towards becoming placenta cells.
It enhances their capacity to do so, yes.
Wow.
They also found it helped promote the proper formation of blastoids. These are lab-grown structures mimicking the early embryo and encourage the maturation of the trophectoderm cells within those blastoids.
That is different. In mice, it seemed more about maintaining pluripotency or making germ cells, but here it's pushing towards the placenta lineage.
It's a key distinction, highlighting potential species-specific roles. And here's another twist. Unlike the mouse studies where IKG strongly influenced histone demethylation, this study found that adding IKG to human NESCs didn't really change global histone methylation levels much.
No change in methylation. So what is it doing in the human cells then?
Instead, they found that higher IKG levels led to a decrease in the availability of another key metabolite, acetyl-CoA.
Acetyl-CoA. Okay, remind us why that's important here.
Acetyl-CoA is another central metabolic hub, but critically, it's the essential cofactor for histone acetyltransferases, or HATs. These enzymes add acetyl groups to histones.
Okay, and what does acetylation do if methylation is a dimmer switch?
Acetylation is generally like turning the lights on. It tends to open up the chromatin structure, making genes more accessible and active. It's strongly associated with maintaining pluripotency, keeping those stem cells in their flexible state.
So if a psyche goes up and acetyl-CoA goes down,
Then you have less fuel for the HAT enzymes, leading to less histone acetylation overall. The researchers proposed that this reduction in histone acetylation weakens the gene network that keeps human NESCs pluripotent, making them more receptive to differentiating.
that's a fascinating mechanism. So in mice, the lever seems to be AKG boosting demethylation to maintain pluripotency, but in humans, it's AKG reducing acetyl-CoA, leading to less acetylation, which helps them leave pluripotency and head towards the trophectoderm.
That appears to be the key difference suggested by this study. It underscores why we need to study human systems directly. The rules aren't always the same.
Absolutely. So sticking with the human study, they also used that modified DM-AKG. What happened when they treated the human NESCs with that?
Consistent with the idea of weakening pluripotency, they found that treating human NESCs with the MAG led to a decrease in the expression of key pluripotency genes, names like KLF17, KLF4, and NANOG. At the same time, they saw a slight increase, a de-repression of early trophectoderm marker genes like GATA3 and CDX2.
So it dampened the stay pluripotent signals and slightly nudged the become placenta signals.
Exactly. But it wasn't a dramatic, complete switch. Their interpretation was that AKG isn't acting as a direct, powerful inducer of trophectoderm fate itself. Rather, it's preparing the cells, making them more competent or ready to respond to the actual signals that will fully push them down that path.
So it's priming the pump, essentially, setting the stage.
That's a good way to put it. It makes them more receptive.
To really nail down this acetylation mechanism, the researchers also inhibited P300. That's a key HAT enzyme, right? One of those acetyl-adding enzymes.
Correct. P300 is a major histone acetyltransferase.
So what happened when they blocked it?
This was a really neat experiment. They found that inhibiting P300 directly, which obviously reduces histone acetylation, produced a very similar outcome to treating the cells with DMKG.
Really, the same effect.
Very similar. They saw repression of those same pluripotency genes, and importantly, the cell showed an increased competence to be induced towards the trophectoderm lineage.
Okay, that strongly suggests the effect of AKG is running through this acetylation pathway.
It provides strong support for that mechanism, yes.
Yeah.
The reduction in histone acetylation seems to be a crucial step in how elevated AKA promotes the trophectoderm fate in these human-naive cells.
It all ties together quite nicely. Higher AKG, lower acetyl-CoA, less HT activity, lower histone acetylation, weakened pluripotency, increased readiness for trophectoderm.
That seems to be the working model based on their findings.
Okay, one last bit from that study. They looked at blastoid formation, those mini embryo models. How did AKG pretreatment affect them?
The results there were also quite telling. Blastoids grown from NESCs that had been pretreated with AKG were noticeably larger.
Larger. Interesting.
And importantly, they showed a really robust induction of cells expressing GATA3, that trophectoderm marker, while still maintaining MANOG expression, the pluripotency marker in the inner cells, mimicking the inner cell mass. So the basic structure was there, but the trophectoderm seemed enhanced.
And was it just more TE cells, or were they different?
That's what they looked at next, using single-cell RNA sequencing. This lets you see the gene expression profile of individual cells. They found that the trophectoderm-like cells in the IKG-treated blastoids were expressing markers associated with mature polar trophectoderm.
Polar trophectoderm, what's that?
That's a specific subtype of trophectoderm in the natural blastocyst that directly contacts the inner cell mass and plays a key role in implantation. So the finding suggests IKG doesn't just initiate TE fate, but also promotes its maturation towards a more functional state within that blastoid structure.
So it's not just quantity, but also quality or maturity of the placenta precursor cells. That's quite something. Okay, taking all this incredible work together, the mouse studies, the human studies, the metabolic links, the different epigenetic mechanisms, what are the big unanswered questions still lingering? Where does the research go next?
That's the crucial question, isn't it? Despite all this progress, there are still major gaps. A big one, as the EMBO article highlighted, is the need for more in vivo studies. Understanding how AKG and other metabolites function within the complex dynamic environment of an actual developing embryo is key. We do a lot in dishes, but the body is different.
Right, moving from the lab bench to the real thing.
Exactly. And related to that is understanding the influence of external factors. How does maternal diet, for instance, impact these crucial metabolite levels in the embryo or germ cells? How do environmental exposures play in? We lack good ways to measure how diet translates to specific metabolite levels inside these tiny early cells.
And how sensitive are those epigenetic enzymes to small changes in metabolite levels? Is there a threshold?
Another excellent question. We don't really know the dose-response dynamics within the cell. And what about long-term effects? Do these metabolite-influenced epigenetic changes stick around? Could they even have effects across generations? We're just scratching the surface there.
Plus the species differences we saw.
Absolutely. The contrasting roles in mouse versus human cells really underscore that we can't just extrapolate. We need to understand the specific regulatory networks in humans. How is metabolite signaling integrated differently?
So lots of questions. How do we tackle them?
Well, the good news is the tools are getting better all the time. Genome editing technologies like CRISPR allow us to precisely manipulate genes involved in metabolic pathways or epigenetic regulation. And advances in single-cell multi-omics, looking at RNA, proteins, epigenetics, and hopefully soon metabolites, all in the same single cell, offer incredible resolution. These technologies hold huge promise for untangling this complexity.
It sounds like a field ripe for more discovery.
Definitely. Understanding the interplay between different metabolic pathways, not just AKG in isolation, and how they collectively fine-tune cell fate is probably the next major frontier.
This deep dive into alpha-ketoglutarate and stem cell fate has been, well, truly illuminating. It reveals this incredibly intricate dance between how a cell makes energy and manages its building blocks, and its very identity, what it is and what it can become. From ROCK-EG's role in mouse cells, perhaps maintaining flexibility or guiding germ cell fate, to its different job in human cells, nudging them towards the placenta by tweaking acetyl-CoA levels and histone acetylation. It's clear this small molecule is a critical conductor in the symphony of early development. The contrasts between species are a fantastic reminder of biology's complexity and why this research, especially in human systems, is so vital. Unraveling these mechanisms fully is still a work in progress, but the potential implications are huge for understanding how life starts, for regenerative medicine, maybe even for fertility and healthy pregnancies. Thinking about how we might one day use metabolic signals to guide cells. That's really exciting stuff. Thank you.